Assalamualaikum, I am Siti Zulaikha binti Nasir. I'm going to present on the topic which I have done for my FRP project, which is synthesis of Goniodiol by using catalytic asymmetric allylation heterodiols allylation approach. Without further ado, let's start with the first chapter, which is the introduction. Why do this study was performed at first? This is because it's related to the natural products, and natural products happen to help lots of people in curing their diseases. However, it is so hard to perform a modification due to its complex structure and also a limited supply of the natural products. That is why it is necessary for the researcher to come up with a solution in order to overcome the limited supply of these natural products and also to make the modification possible. So the modification is actually necessary in order to enhance the therapeutic use of the natural products. There are actually many substances in the natural products that help in curing the diseases, which one of it is the stereolactones. And this is the focus of this study, which is the stereolactones and more focusing on Goniodiol. So what would be the benefits of this study? So one of it is it will be beneficial to society because it will help to cure more diseases. Next, it would also help the researcher in providing more ways in synthesizing Goniodiol, which could also be beneficial in the future. So the objective of the study is to synthesize the intermediate of the Goniodiol and also to elucidate the structure by using carbon and also proton NMR spectroscopy. Moving on to the next chapter, which is the literature review. So Goniodiol is a stereolactones usually being found in a species called Goniotalamus. Goniotalamus could also possess cytotoxic activities, which coming from the compound called stereolactones. So one of the stereolactones, it is Goniodiol. But however, Goniodiol were only found is Goniotalamus giganteus, Goniotalamus ammoion, Goniotalamus graffiti, and also Goniotalamus sesquipedalis. Research gap here is clearly proven that Goniodiol has a limited natural distribution. So that is why more synthesis is needed. So one of the approaches of synthesizing Goniodiol is called stereoselective. So stereoselective, it would selectively choose in which site that they wanted to perform the reaction, and it would convert the prochiral group of it in order to get the products. So the next approach is called the diastereoselective method, and the article that was chosen, it involves two types of mechanism, which is the Morita-Bellis human followed by the diastereoselective. So the Morita-Bellis human is necessary to produce the stereolactones. The stereolactones produced from this study was leocarbonate. And it was found out that the same intermediate could also be used to synthesize the Goniodiol. So the next approach are called Carrera alkylation and also the sharpless asymmetric method. So these two is actually the mechanism used in order to just performing the intermediate of the Goniodiol. And the last approach are called the co-catalyzed hydrolytic kinetic resolution, which this method was used in order to convert the sin-alkoxy epoxide into etiols and also epoxide with two stereocenters. And more mechanism is needed in order to perform reaction to create the homoallylic alcohol. Only then Goniodiol would be synthesized. Up next is the methodology. First thing that was done is the preparation of the triisopropoxy titanium chloride. The first thing that was done is the dilution of the titanium tetrachloride. This is because the available reagent happened to be very concentrated. This is because the concentration was 1 mole. The concentration proposed by the article was 0.12 millimole only. So after the dilution, only 2.4 ml of it was needed, and 110 microliter of titanium isopropoxide was added to it. After the mixture was set aside for about 1 hour, 56 mg of silver oxide was added to it. It was stirred for about 5 hours with no direct light. It was then diluted with dichloromethane, and after that, carbinol was added, and it was set aside for about 2 hours. Mixture was then frozen to negative 15 degrees Celsius before adding the transenamorthyhide and also the allyl tributyltin. The mixture was let to be in the temperature of 0 degrees Celsius for approximately 36 hours. Then it was quenched, extracted, and also purified by column chromatography. TLC was done to the product and also the NMR spectroscopy. Moving on to the results and also discussion. So the general idea of this reaction is that it requires an allyl tributyltin and also aldehyde to produce the homoallylic alcohol. It would possibly happen is by the help of a Lewis acid catalyst, and the most suitable Lewis acid catalyst to be used is the B-tip complex catalyst. So B-tip stands for Binol and Titanium complex catalyst. It actually helps in activating the aldehyde and then followed by the attachment of the allyl ligand from the allyl tributyltin. So that is basically the reaction that happens in the study. The first result obtained was from the TLC of the first trial. So the results obtained before the column chromatography, it was observed that there were 5 spots that were spotted on top of the TLC plates, which is the product, before letting it to be cooled down for 36 hours and after it. And then we have the aldehyde, the allyl tributyltin, and also the catalyst. As for the first product, before letting it cool down, 4 spots were observed, which it belongs to the allyl tributyltin, the aldehyde, and also the catalyst. And there were 1 spot that is claimed to be the unknown spot, and it was predicted to be the desired product. But after letting the sample to be cooled down, there were no longer unknown spots left. Column chromatography was still done in order to purify the sample. So that comes up the result of the after column chromatography. There were only spots that were spotted on top of the TLC plate. Surprisingly, there were only 2 spots left for the product. So there is a possibility that the reaction was not performed yet, but still, the product was sent to the NMR laboratory to confirm it. Next is the proton NMR spectrum obtained for the first trial. And it was compared to the spectral data analysis that was mentioned in the article. None of it that is close to each other. But when it was compared to the proton NMR spectrum of the transenamyaldehyde and also allyl tributyltin, both of these NMR spectrum somehow fits the shape of the peaks obtained in this proton NMR spectrum. So it can be said that allyl tributyltin and aldehyde was there in the product, but however they did not perform the reaction. So from the first trial's result, it can be seen that there are some of the procedures that need to be improvised to complete the reaction. So initially, the methodology that was mentioned by Connie was followed because the condition and also the temperature was somehow easier to be followed. So by that, the procedure was switched to D. Fatima's methodology, and the concentration of the titanium tetrachloride was also diluted. Up next, these are the 2 TLC trials obtained after repeating the procedure back again, but followed the D. Fatima's methodology. After the sample had been cooled down, the unknown spots remained, but other spots had gone, and the same goes to after column chromatography. It was predicted that the reaction has been completed. This is how the spectrum for the proton NMR and also the carbon NMR looks like. When it was compared to the literature data, every peaks mentioned was there in the spectrum. Since the structure of the gonodiol has 12 carbons, so the carbon NMR spectrum should have 12 peaks that were observed. Yes, all of the 12 peaks was there. The first peaks that wanted to be observed is that the proton in the benzene ring. So there are peaks shown which in the range of 7.43 to 7.26 ppm. Another most important peaks to be observed is the one that attached directly to the oxygen, and there was actually a peak shown for this, which is the 1.93 ppm. Other than those important peaks, there were also other peaks that were observed in this proton NMR spectrum. All of it has 1 integration. So the percent yield obtained for this experiment was only 1.05%, which far more or less as compared to the one reported in the literature data. Throughout the study, intermediate gonodiol was successfully yielded with the percent yield of 1.05%, and the structure of the intermediate gonodiol was successfully elucidated. As for the recommendation, the first thing that needs to be improvised is that to follow the method precisely. And the second one is to let the product to settle down a bit longer in order to complete the reaction. The third one is to obtain a better and cleaner baseline resolution for the spectrum is by purifying the product even more. The last one is to repeat the procedure even more, obtain a better percent yield as compared to before. So that's all for me. Thank you.